Hello. Hi, and thanks for taking a look at this little video that I was asked by the graduate school to record instead of the usual welcome speech in Reitenhaslach, which I would have liked to do to meet you in person, but which during these times, unfortunately, is not possible. So instead, I'm saying hi from my home here in the area of Freising, and I hope that you will like this little video anyway. I called it with a little help from my friends, and you will find out why. But very broadly speaking, this is about what I would like to talk to you about, how a little help can help to really improve your life. And so my name is Philip Benz. I'm a professor at TUM since 2014. My professorship is called Wood Bioprocesses, which, however, means that we're fungal geneticists and biotechnologists. But don't worry, you do not have to be a biologist to understand this video, or if you're not, you would get lost. That's definitely not the case. I hope this is for everybody and is more based on my life experience. But you will have to excuse me if I'm using the fungi, which is what we're the model system that we're working on, to explain a few of these concepts that I would like to talk to you about. So let's first show you a little bit about my live station so that you just know who's talking to you. When you can see me up there in the video, but also down there, this is how it usually looks like when I'm giving this talk, or something like this talk in Reitenhaslach. So I started to study biology in Braunschweig, but then I had two stays in the US, one for my diploma thesis in San Diego and one for my postdoc, where I really started to go into fungal genetics in Berkeley. And in between, I did my PhD at the other Munich university at the LMU, which that was still in plant biochemistry. So I'm a plant and fungal person. And here's one little sidetrack that I would like to take to give you a very, very good hint. And this is to go away. And what I mean with this is I'm very happy for all of those of you who come from abroad and now have joined TUM. But those of you who are German and who have studied in Germany and will also do their PhD in Germany, I really recommend to do your postdoc later abroad, or choose a possibility that also the grad school can help you with, to go abroad for a few months, because it really is not only very nice to go abroad, and this is just an example, the view that I had over the town of Berkeley and the Golden Gate Bridge in the background. So you do not just only visit nice places when you're abroad, but you also get an outsider's view of your own country, which can be quite healthy in many aspects, and was very enriching, at least for me in my life. So definitely consider that if you have the chance. So after these first words, welcome to TUM again. I'm really happy that you made the decision to join this university, and I welcome you to your doctorate. And so I've obviously also gone through this process, and when I was asked to record a little video for you as an introduction, I thought about what happened during my doctorate, and so what's probably in store for you. I was wondering, well, what is lying ahead of you? And what I really wish for you is that you will have the possibility to do great science, to meet fantastic colleagues, to write impactful papers, and in some, to just have a great time. But I guess it's also okay to be a little bit more realistically. So what's probably in store for you is a great amount of work, hopefully fantastic work and intriguing work, but there will be a great amount of work. Many more questions than answers, and this is true for basically all studies that people do. And a supervisor with probably very little time, and I know what I'm talking about, even though I'm not so long a professor. And then maybe when you start writing it all up and you're really, really amped up, you will hit a writer's block and everything will go down the drain. I really don't hope that this will be the case, but this, out of experience, this might happen. So also out of my experience, I've seen people that approach this in two different ways, mostly. Either there's the people that are really outgoing, they communicate their problems and they discuss with their peers and their bosses the problems and find a solution together, or they keep just pushing forward, trying to make this experiment work that doesn't want to work, on all costs, and work really their asses off, if you excuse this expression. Both of these things I have also done in my life, and I'm here to advocate the first version, which is really, really helpful. So basically, I'm already taking away the bottom line of this video: don't be a loner. But let's take a look at how fungi can maybe help us to understand why this can really be helpful, and how communication and working together and asking for help can be helpful. And I think you will find this also quite interesting. So can fungi help us to see the benefit of this approach? And this is a little friend of mine that used to live in my backyard, Tramedes, on a cherry tree. And so I asked him, and I think he was, in favor of this, and he was pretty happy to help us. So let's look into this in a little bit more detail. So clearly, you are here to do great things, and we want you to do great things. And fungi are a good example because they can do great things. Not sure if you were aware of this, but the greatest thing, the greatest organism on this planet is not the blue whale or the elephant. It's actually a fungus. And so this made the news a few years back. And this is an area on the size of more than 1,300 UEFA-sized soccer fields in Oregon. And so all the samples of soil, or where this Armillaria mushroom here grows, all the samples of this mushroom that were harvested in this area showed that it's actually one and the same organism. So it's not really visible unless it's fruiting, but the soil is full of just one organism, and you're treading on this over really hectares and hectares of area. Considering that 1 gram of soil can have 60 kilometers of fungal hyphae sitting in it, you get a feeling of how much biomass that really is that is distributed there. Also, you can do great things with fungi. Not only fungi can do great things, but you can also work with them to do great things. So if you take agricultural waste, for example, and then you have some of these fungi grown and they make these nice hyphal nets, mycelia, then you can press this into mushroom material. And this is actually quite, on the forego these days. And there's these art projects where people have really made entire structures and buildings just with fungal material. You can even work on it and make it look, feel, even taste, I guess, but be almost like leather that has the same characteristics. So the way this fungal mycelium is made is so strong that you can do these great things with it. But there's a few things that are necessary to make it so strong. And also, spores in the beginning, fungal spores, what every fungal starts off with, are very tiny. So kind of like us at the beginning of our career, we start at humble beginnings, but we want to do great things. So we have to develop ourselves, and fungi also have to develop. So fungi start out as spores, micrometer size, tiny but many. And somehow these many hyphae that come out of these spores would have to make this really strong net. And what is important to make this net is that they cross-connect. And so that's not so easy, because for this, hyphae have to find each other, they have to meet each other, they have to understand if they are compatible with each other or not, and then they have to fuse. And this is something that's very important for fungal development, and that is important then to make these hyphal nets that we have already seen. So many single hyphae plus connections make a mycelial net. If they were not able to do that, they could not be able to make a strong organism or a strong tissue in a way. So that's really, really important. And they do that really by communicating, if you believe it or not. I was very astonished also when I saw this video the first time. This is why I would like to show that to you. This is showing that fungi clearly communicate with each other. So they're doing that, what I already mentioned in the beginning. It's really good to talk to each other. already at this, very teeny tiny stage. So what you see here are two spores of the fungus that we like to work with in the lab as well, in the Rossbach crassa. And the video is from a friend of mine in Braunschweig who's working with the same organism. And he is studying exactly this communication. So what you see is that these two hyphae are germinating out, and all the biologists among you would already saw that there's a protein labeled with a green fluorescent protein here. So you can see when it is made and where it is made in these fungi. And obviously it's being made at the tip of these fungi. And you can see very strongly here and very weakly here. But now observe what's happening. Because the signal is going away here and it's getting much stronger here. And then it's going away here and it's coming back on this one. So obviously they are shouting out something and now they're finding each other. And this protein that is oscillatingly on and off in these hyphae is involved in this communication. And it's really like an I call, I listen, I call, you listen. And now they find each other. There's a final kiss. They both light up at some point, and then they fuse, which is now the case. So this is pretty extraordinary already by itself. But really what this means is that fungi that at this point are really unicellular organisms can already perform a communication that is very, very similar to what we are doing. Yeah? When I'm talking, usually in a live situation, not now, but when I'm talking, you're listening, and then if you have a question, I'm shutting up to wait for your question, and then I'm answering again. If we both would talk at the same time, there's no communication. So communication is something that's really, really basic, even for these kind of organisms, very, very, small microorganisms, but it helps them to do great things. So maybe realizing that they're doing stuff that is very similar to what we are doing is a little bit less surprising if we look at the tree of life. So this is where we animals are, and this is where the plants are, and to this area the fungi had been grouped for the longest time of academic history. But now we know that fungi are actually a side branch of something that also goes to us animals. So next time when you go to the forest, really reconsider, because every fungus that you will meet, every mushroom that you will see is so much more related to you than it is to the plants that are sitting next to it, that it's really astonishing. So, treat them with care. All right, so we have seen fungi communicate and it helps them to do great things, but where do they get all the energy for this from? I mean, if they're doing great things, that also takes a great amount of energy. And this comes very close to what we are studying in my lab. Well, maybe we can start with this picture. So take a look at this. What you can see is a forest. This is a picture out of the Bavarian Park National Forest, and all you can see is trees. And you could take a bit of this wood and chew on it, and it would just taste like wood, but not sweet. But the fungi actually see this world differently, because if we look through the world of the fungi, and this is kind of illustrated by this, they can see what the wood is actually made of. And wood is mostly made of cellulose, and cellulose is the main ingredient of all plant cell walls. It's actually long chains of glucose, and that's nothing else than the household sugar that we're also using at home, but connected in a way that we cannot access it. It's really, really strong, or otherwise trees would dissolve in the next rain, and they don't. It's really, really strong, and we cannot break this down, but the fungi can. And this is something that we in my lab also want to study, or that we are actively studying, and try to apply this in biotechnological applications. So let's take another example where you can see that they are really able to do this very, very greatly. So let's take a book. And this is a few still pictures from one of the videos that the Cornell Plant Pathology Photo Lab has posted on YouTube, which I really recommend to check out. But if you take a book that's really made only of paper, and paper is pure cellulose, and you inoculate that with fungal spores, and of course you keep it moist, after about 1 month, a little bit more than 1 month, this is what you see. So you can, I'm not sure if in your life you have ever bitten in a book, but this was never sweet, I'm sure, in your mouth. But the fungi can really break it down and make a living of it. And this is something that they can do really well, and we want to understand why. And it's also very important for our next point that I would like to make. This is, first of all, another representation of how cellulose looks like. Cellulose is also a net, but a net of sugars. And these are these cellulose fibrils that are made of microfibrils and nanofibrils. And basically, these are all in their inner core made of these long chains of glucose, as I mentioned. And these sugar molecules itself are very tasty also for us. But what is difficult for us to break is this little glycosidic linkage. It's very similar to the one in starch, because starch is also a long chain of glucose, but this linkage is just ever so slightly different, and we can break it. But this beta 1,4 glycosidic linkage is inaccessible for us, but the fungi can do this. So they are able to break down this really, really crystalline molecule of cellulose. This is how it looks in a realistic modeling. When you pack all of these strains together, you can see how close and tight this is, and it becomes really insoluble and very difficult to attack for enzymes. But they have somehow found a way. So this is similar to a problem that also you might approach in your PhD, yeah? Something that's a really hard nut to break. So how would you think about this? Well, you could say, leave me alone. This is just one insoluble problem more on my way to a PhD. And you can look for one of these very nice PhD comics. Then there's pretty much one for every situation in your life. Maybe that cheers you up, but it doesn't really help, I guess. Or you can start becoming creative. So the way that microorganisms like fungi came about solving this problem was that they said, well, if there's not one enzyme that can do it, let's maybe try it with two, and they help each other. And by helping each other, they create synergy. And this is exactly what I would like to show you. So let's take a look in a little bit more detail. These are the glucose strands again, and now I would like to introduce these little enzymes that are doing the job. Since long strains of glucose are called glucans, the enzymes that degrade it are called glucanases. And there's these two versions of these enzymes. Both are highly related to each other, but have slightly different functions. The one are exoglucanases, and these guys can produce a lot of product. They can really digest cellulose, but they need an end of the chain in order to attack. So they need an end, and then they can play the Pac-Man and start chewing and releasing these sugar molecules. But without an end, they can't do anything. And then there's these endoglucanases. They don't need an end. They can bite in the middle of the chain, randomly, and then they will let go again. They don't really produce a lot of products, but they produce ends. And so with these ends, more exoglucanases can bind and can produce more products. And this together creates synergy. This is a little bit abstract, I know, and usually what I do with you guys is I get a few of you on stage and we play endo and exoglucanases, and we digest these long strains of glucose, which are these colorful flags. Most of you guys play exoglucanases, and one gets this nice vest and plays an endoglucanase, and then we can see how this changes the dynamics. Unfortunately, this is not possible today. So I thought about how to, come up with something else, and I talked to my daughters, and they actually helped me to make a stop-motion movie. And in come the main players of this movie. There's little Aldi emojis, Rocky, Lolo, Oopsie, and Happy, as well as Bling Bling. These yellow guys are the exoglucanases, and Bling Bling is the one endoglucanase. They're very similar, as they are also in nature, but they have slightly different functions, as I explained, and as we'll also see in this movie now. So let's take a look. All right, so in come our friends, the exoglucanases. Rocky, Lolo, Oopsie, and Happy. And so they are looking for ends, but this glucan chain here made of sugar crystals only has two ends. So only these two guys can start and become active, while all the others, or these two others, at least in this case, in this example, are bored and they can't really do anything. Yeah, so they like ends and they work, but they're slow. And this is also what the text will say. But you can also nicely ignore this and just listen to me. But yeah, this is not very efficient. Alone by themselves, they cannot do much. So they decide to call their friend, and they call an endoglucanase Bling Bling. Here comes in Bling Bling. They're very happy to greet their friend. Hello. And now Bling Bling, as an endoglucanase, can start to attack, site open or not open, closed sides of that chain and producing more openings there, which are immediately attacked by his yellow friends. And they can start becoming very active there. And by now, he's creating so many ends that there's actually not enough exoglucanases, so we had to bring in another friend, Ghostly, here. And now these guys are really, really active. They all can produce at their highest efficiency, and the sugar strand is being taken down very quickly. They can do more together, and this is called synergy, the exo-endo synergy. So let's just wait for them to do their job, which is much faster than before. And now they greet each other one more time, and you can see all the bunch of sugar that they have produced. So we can also look at this a little bit more theoretically. Just very briefly, if you only take exoglucanases, as we have seen, that would be the green line, or you could only take endo enzymes and you have them digest cellulose. This is the low activity that you get, so the amount of sugar molecules being released from the chains. And if we then put them together and have them work together, you get a additive activity. If you just calculate this plus this is what you would expect to get. But really what you get in real life is this. And so the difference between the calculated activity and that what you really get, that's called synergy. You get the real efficiency gain. So what have we learned? We have learned you don't have to be specialized in everything. It's beneficial sometimes to distribute the workload, which does not mean that you need to find somebody for each and every work package of your work. But sometimes you really have a tight problem and a big problem. It might be efficient to ask somebody to help you with a little bit. May it be just by some advice or by doing a little piece of work for you, which brings you a lot further. And very often it's really the case that it doesn't need a lot of help to get you further. I've experienced this myself. After having worked on a problem for many months and I couldn't solve it, I just talked to a colleague and explained to him my problem, and he was like, well, but this is all very interesting, but did you know that so-and-so? And I could only go like, no, I have no idea about that, but I'm going to look into this. And it was really a breakthrough moment because there was some information that I just didn't have, and then he had gotten by chance that helped me to make huge strides in this project in no time, after having sitting on it for long months without getting any further. So in the end, the essence of synergy is the result is more than the sum of the parts. So there can be very little help that produce a lot of gain in the end. So our take-home messages for today: fungi are much closer to humans than one would have thought, and they're able to do great stuff, and humans are also able to do great stuff with them. And so they're a pretty good example of the great stuff that you hopefully are going to make during your doctorate. What the fungi have been particularly good at in this video, but also they are every day in the world out there, is that they can break the backbone of sugar polymers. Cellulose being one of the most abundant of those out there. All plant biomass is made of this, but it's also the hardest nut to crack. And they have made this solution that they create synergy by helping themselves, or getting two different enzymes to help themselves, and that has a big effect. And this is also what I would like you to do. Use your time at the university, talk to people, talk to colleagues, talk to friends, tell them about your cool stuff that you're doing, but maybe also about your problem. Don't hide them. And if you help each other, you might get that little bit of help that you need to really get you a long stride forward. And this is really what I would like to give you on your way. So with this, I would like to thank, of course, my lab that has helped me, all the research that also, in an abstract way, flew into this video. Our funders, the money givers, of course, all the tax dollars, or the tax euros in this case, here in Europe, that also flow into this. So all of you for paying your taxes, that also funds our research. And of course, thank you for your attention on this video. If you ever want to talk to me, ask me something, there's my email address. You can also find me online. I'll be here for a little bit, and I hope that you will be too. Bye-bye.